Chapter 11 Membrane Structure. A living cell is a self-reproducing system of molecules held inside a container. That container is the plasma membrane, a fatty film so thin and transparent that it cannot be seen directly in the light microscope. Every cell on Earth uses a membrane to separate and protect its chemical components from the outside environment. Without membranes, there would be no cells, and thus no life. The plasma membrane is simple in form. Its structure is based on a 2-ply sheet of lipid molecules about 5 nanometers or 50 atoms thick. Its properties, however, are unlike those of any sheet of material we are familiar with in the everyday world. Although the plasma membrane serves as a barrier to prevent the contents of the cell from escaping and mixing with the surrounding medium, Figure 11.1a, it does much more than that. If a cell is to survive and grow, nutrients must pass inward across the plasma membrane, and waste products must pass out. To facilitate this exchange, the membrane is penetrated by highly selective channels and pumps, protein molecules that allow specific substances to be imported and others to be exported. Other proteins in the membrane act as sensors that enable the cell to receive information about changes in its environment and respond to them. Figure 11.2. The mechanical properties of the membrane are equally remarkable. When a cell grows or changes shapes, so does its membrane. It enlarges in area by adding new membrane without ever losing its continuity, and it can deform without tearing. If the membrane is pierced, it neither collapses like a balloon nor remains torn. Instead, it quickly reseals. The simplest bacteria have only a single membrane, the plasma membrane. Eukaryotic cells, however, also contain an abundance of internal membranes that enclose intracellular compartments to form the various organelles, including the endoplasmic reticulum, Golgi apparatus, and mitochondria. Figure 11.3. These internal membranes are constructed on the same principles as the plasma membrane, and they too serve as highly selective barriers between spaces containing distinct collections of molecules. See Figure 11.1b. Subtle differences in the composition of these membranes, especially in their resident proteins, give each organelle its distinctive character. Regardless of their location, all cell membranes are composed of lipids and proteins and share a common general structure. Figure 11.4. The lipids are arranged in 2 closely opposed sheets, forming a lipid bilayer. See Figure 11.4b and c. This lipid bilayer gives the membrane its basic structure and serves as a permeability barrier to most water-soluble molecules. The proteins carry out most of the other functions of the membrane and give different membranes their individual characteristics. In this chapter, we consider the structure of biological membranes and the organization of their 2 main constituents, lipids and proteins. Although we focus mainly on the plasma membrane, most of the concepts we discuss also apply to internal membranes. The functions of cell membranes, including their role in cell communication, the transport of small molecules, and energy generation, are considered in later chapters. The lipid bilayer. The lipid bilayer has been firmly established as the universal basis of membrane structure, and its properties are responsible for the general properties of all cell membranes. Because cells are filled with and surrounded by solutions of molecules in water We begin this section by considering how the structure of cell membrane is a consequence of the way membrane lipids behave in a watery, aqueous environment. Membrane lipids form bilayers in water. The lipids in cell membranes combine two very different properties in a single molecule. Each lipid has a hydrophilic, water-loving head and one or two hydrophobic, water-fearing hydrocarbon tails. Figure 11.5. The most abundant lipids in cell membranes are the phospholipids, molecules in which the hydrophilic head is linked to the rest of the lipid through a phosphate group. The most common type of phospholipid in most cell membranes is phosphatidylcholine, which has the small molecule choline attached to a phosphate as its hydrophilic head and two long hydrocarbon chains as its hydrophobic tails. Figure 11.6. Molecules with both hydrophilic and hydrophobic properties are termed amphipathic. This chemical property is also shared by other types of membrane lipids, including the sterols, such as the cholesterol found in animal cell membranes, and the Glyco Lipids, which have sugars as part of their hydrophilic head. Figure 11.7. Having both hydrophilic and hydrophobic parts plays a crucial part in driving these lipid molecules to assemble into bilayers in an aqueous environment. As discussed in Chapter 2, hydrophilic molecules dissolve readily in water because they contain charged atoms or polar groups. That is, chemical groups with an uneven distribution of positive and negative charges. These charged atoms can form electrostatic attractions or hydrogen bonds with water molecules, which are themselves polar. Figure 11.8. Hydrophobic molecules, by contrast, are insoluble in water because all or almost all of their atoms are uncharged and nonpolar. They therefore cannot form favorable interactions with water molecules. Instead, these nonpolar atoms force adjacent water molecules to reorganize into a cage-like structure around the hydrophobic molecule. Figure 11.9. Because the cage-like structure is more highly ordered than the surrounding water, its formation requires energy. The energy cost is minimized, however, if the hydrophobic molecules cluster together, limiting their contact with the water to the smallest possible number of water molecules. Thus, purely hydrophobic molecules, like the fats found in animal fat cells and the oils found in plant seeds, figure 1110a, coalesce into a single large drop when dispersed in water. In contrast, amphipathic molecules, such as phospholipids, figure 1110b, are subject to two conflicting forces. The hydrophilic head is attracted to water, while the hydrophobic tail shuns water and seeks to aggregate with other hydrophobic molecules. This conflict is beautifully resolved by the formation of a lipid bilayer, an arrangement that satisfies all parties and is energetically most favorable. The hydrophilic head faces the water from both surfaces of the bilayer sheet. The hydrophobic tails are all shielded from the water as they lie next to one another in the interior, like the filling in a sandwich. Figure 11.11. The same forces that drive the amphipathic molecules to form a bilayer make the bilayer self-sealing. Any tear in the sheet will create a free edge that is exposed to water. Because this situation is energetically unfavorable, the molecules of the bilayer will spontaneously rearrange to eliminate the free edge. If the tear is small, This spontaneous rearrangement will exclude the water molecules and lead to repair of the bilayer, restoring a single continuous sheet. If the tear is large, the sheet may begin to fold in on itself and break up into separate closed vesicles. In either case, the overriding principle is that free edges are quickly eliminated. The prohibition on free edges has a profound consequence. The only way a finite sheet can avoid having free edges is to bend and seal, forming a boundary around a closed space. Figure 11.12. Therefore, amphipathic molecules such as phospholipids necessarily assemble into self-sealing containers that define closed compartments. This remarkable behavior, fundamental to the creation of a living cell, is in essence simply a result of the property that each molecule is hydrophilic at one end and hydrophobic at the other. The lipid bilayer is a two-dimensional fluid. The aqueous environment inside and outside a cell prevents membrane lipids from escaping from the bilayer, but nothing stops these molecules from moving about and changing places with one another within the plane of the bilayer. The membrane therefore behaves as a two-dimensional fluid, which is crucial for membrane function and integrity. Movie 11.1. This property is distinct from flexibility, which is the ability of the membrane to bend. Membrane flexibility is also important, and it sets a lower limit of about 25 nanometers to the size of vesicle that cell membranes can form. The fluidity of lipid bilayers can be studied using synthetic lipid bilayers, which are easily produced by the spontaneous aggregation of amphipathic lipid molecules in water. Two types of synthetic lipid bilayers are commonly used in experiments. Closed spherical vesicles, called liposomes, form if pure phospholipids are added to water. They vary in size from about 25 nanometers to 1 millimeter in diameter. Figure 11.13. Alternatively, flat phospholipid bilayers can be formed across a hole in a partition between two aqueous compartments. Figure 11.14. These simple artificial bilayers allow measurements of the movements of the lipid molecules, revealing that some types of movement are rare while others are frequent and rapid. Thus, in synthetic lipid bilayers, phospholipid molecules very rarely tumble from one half of the bilayer or monolayer to the other. Without proteins to facilitate the process and under conditions similar to those in a cell, it is estimated that this event, called flip-flop, occurs less than once a month for any individual lipid molecule. On the other hand, as a result of thermal motions, lipid molecules within a monolayer continuously exchange places with their neighbors. Figure 11.15. This exchange leads to rapid diffusion of lipid molecules in the plane of the membrane so that, for example, a lipid in an artificial bilayer may diffuse a length equal to that of a large bacterial cell, about 2 micrometers, in about 1 second. If the temperature is decreased, the drop in thermal energy decreases the rate of lipid movement, making the bilayer less fluid. Similar results are obtained when one examines isolated cell membranes and whole cells, indicating that the lipid bilayer of a cell membrane also behaves as a two-dimensional fluid in which the constituent lipid molecules are free to move within their own layer in any direction in the plane of the membrane. These studies also show that lipid hydrocarbon chains are flexible and that individual lipid molecules within a monolayer rotate very rapidly about their long axis. some reaching speeds of 30,000 revolutions per minute. See Figure 1115. In cells, as in synthetic bilayers, individual phospholipid molecules are normally confined to their own monolayer and do not flip-flop spontaneously. The fluidity of a lipid bilayer depends on its composition. The fluidity of a cell membrane, the ease with which its lipid molecules move within the plane of the bilayer, is important for membrane function and has to be maintained within certain limits. Just how fluid a lipid bilayer is at a given temperature depends on its phospholipid composition and, in particular, on the nature of the hydrocarbon tails. The closer and more regular the packing of the tails, the more viscous and less fluid the bilayer will be. Two major properties of hydrocarbon tails affect how tightly they pack in the bilayer: their length and the number of double bonds they contain. A shorter chain length reduces the tendency of the hydrocarbon tails to interact with one another and therefore increases the fluidity of the bilayer. The hydrocarbon tails of membrane phospholipids vary in length between 14 and 24 carbon atoms, with 18 to 20 atoms being most usual. Most phospholipids contain one hydrocarbon tail that has one or more double bonds between adjacent carbon atoms, and a second tail with single bonds only. See Figure 11.6. The chain that harbors a double bond does not contain the maximum number of hydrogen atoms that could, in principle, be attached to its carbon backbone. It is thus said to be unsaturated with respect to hydrogen. The fatty acid tail with no double bonds has a full complement of hydrogen atoms. It is said to be saturated. Each double bond in an unsaturated tail creates a small kink in the hydrocarbon tail. See Figure 11.6, which makes it more difficult for the tails to pack against one another. For this reason, lipid bilayers that contain a large proportion of unsaturated hydrocarbon tails are more fluid than those with lower proportions. In bacterial and yeast cells, which have to adapt to varying temperatures, both the lengths and the unsaturation of the hydrocarbon tails in the bilayer are constantly adjusted to maintain the membrane at a relatively constant fluidity. At high temperatures, for example, the cell makes membrane lipids with tails that are longer and that contain fewer double bonds. A similar trick is used in the manufacture of margarine from vegetable oils. The fats produced by plants are generally unsaturated and therefore liquid at room temperature, unlike animal fats such as butter or lard, which are generally saturated and therefore solid at room temperature. Margarine is made of hydrogenated vegetable oils. Their double bonds have been removed by the addition of hydrogen so that they are more solid and butter-like at room temperature. In animal cells, membrane fluidity is modulated by the inclusion of the sterol cholesterol. Figure 11.16a. This molecule is present in especially large amounts in the plasma membrane, where it constitutes approximately 20% of the lipids in the membrane by weight. Because cholesterol molecules are short and rigid, they fill the spaces between neighboring phospholipid molecules left by the kinks in their unsaturated hydrocarbon tails. Figure 11.16b. In this way, cholesterol tends to stiffen the bilayer, making it more rigid and less permeable. The chemical properties of membrane lipids and how they affect membrane fluidity are reviewed in Movie 11.2. For all cells, membrane fluidity is important for many reasons. It enables membrane proteins to diffuse rapidly in the plane of the bilayer and to interact with one another, as is crucial, for example, in cell signaling. Discussed in Chapter 16, it permits membrane lipids and proteins to diffuse from sites where they are inserted into the bilayer after the synthesis to other regions of the cell. It allows membranes to fuse with one another and mix their molecules, and it ensures that membrane molecules are distributed evenly between daughter cells when a cell divides. If biological membranes were not fluid, it is hard to imagine how cells could live, grow, and reproduce. The lipid bilayer is asymmetrical. Cell membranes are generally asymmetrical. They present a very different face to the interior of the cell or organelle than they show to the exterior. The two halves of the bilayer often include strikingly different sets of phospholipids and glycolipids. Figure 1117. Moreover, membrane proteins are embedded in the bilayer with a specific orientation, which is crucial for their function. The lipid asymmetry is established and maintained as the membrane grows. In eukaryotic cells, new phospholipids are manufactured by enzymes bound to the part of the endoplasmic reticulum membrane that faces the cytosol. These enzymes, which use free fatty acids as substrates, see panel 2.4, pages 70 to 71, deposit all newly made phospholipids into the cytosolic half of the bilayer. To enable the membrane as a whole to grow evenly, half of the new phospholipid molecules then have to be transferred to the opposite monolayer. This transfer is catalyzed by enzymes called flipases, figure 1118. In the plasma membrane, flipases transfer specific phospholipids selectively, so that different types become concentrated in each monolayer. Using selective flipases is not the only way to produce asymmetry in lipid bilayers, however. In particular, a different mechanism operates for glycolipids, the lipids that show the most striking and consistent asymmetric distribution in animal cells. See figure 1117. To explain their distribution, it is necessary to take a more detailed look at how new membrane is produced in eukaryotic cells. Lipid asymmetry is preserved during membrane transport. Nearly all new membrane synthesis in eukaryotic cells occurs in the membrane of one intracellular compartment, the endoplasmic reticulum, ER. See figure 11.3. The new membrane assembled there is exported to the other membranes of the cell through a cell cycle of membrane budding and fusion. Bits of the bilayer pinch off from the ER to form small spheres called vesicles, which then become incorporated into another membrane, such as the plasma membrane, by fusing with it. Figure 1119. The orientation of the bilayer relative to the cytosol is preserved during vesicle formation and fusion. This preservation of orientation means that all cell membranes, whether the external plasma membrane or an intracellular membrane around an organelle, have distinct inside and outside faces that are established at the time of membrane synthesis. The cytosolic face is always adjacent to the cytosol, while the non-cytosolic face is exposed to either the cell exterior or the interior space of an organelle. See figure 1119. Glycolipids are located mainly in the plasma membrane, and they are found only in the non-cytosolic half of the bilayer. Their sugar groups are therefore exposed to the exterior of the cell. See figure 1116. Where they form part of a continuous protective coat of carbohydrate that surrounds most animal cells. The glycolipid molecules acquire their sugar groups in the Golgi apparatus. the organelle to which proteins and membranes made in the ER often go next, discussed in Chapter 15. The enzymes that add the sugar groups are confined to the inside of the Golgi apparatus, so that the sugars are added only to lipid molecules in the non-cytosolic half of the lipid bilayer. Once a glycolipid molecule has been created in this way, it remains trapped in this monolayer, as there are no flipases that transfer glycolipids to the cytosolic monolayer. Thus, when a glycolipid molecule is finally delivered to the plasma membrane, it faces away from the cytosol and displays its sugar on the exterior of the cell. See Figure 1119. Other lipid molecules show different types of asymmetric distributions related to other functions. The inositol phospholipids, for example, are minor components of the plasma membrane, but they play a special role in relaying signals from the cell surface to the intracellular components that respond to those signals. Discussed in Chapter 16, they act only after the signal has been transmitted across the plasma membrane. Thus, they are concentrated in the cytosolic half of this lipid bilayer. See Figure 1117. Membrane proteins. Although the lipid bilayer provides the basic structure of all cell membranes and serves as a permeability barrier to the molecules on either side of it, most membrane functions are carried out by membrane proteins. In animals, proteins constitute about 50% of the mass of most plasma membranes, the remainder being lipid plus the relatively small amounts of carbohydrate found on glycolipids and glycosylated proteins. Because lipid molecules are much smaller than proteins, however, a cell membrane typically contains about 50 times more lipid molecules than protein molecules. See Figure 11.4. Membrane proteins not only transport particular nutrients, metabolites, and ions across the lipid bilayer, they serve many other functions. Some anchor the membrane to macromolecules on either side. Others function as receptors that detect chemical signals in the cell's environment and relay them to the cell interior. And still others work as enzymes to catalyze specific reactions. Figure 1120 and Table 11.1. Each type of cell membrane contains a different set of proteins, reflecting the specialized functions of the particular membrane. In this section, we discuss the structure of membrane proteins and illustrate the different ways that they can be associated with the lipid bilayer. Membrane proteins associate with the lipid bilayer in various ways. Proteins can be associated with the lipid bilayer of a cell membrane in several ways. Figure 1121. 1. Many membrane proteins extend through the bilayer, with part of their mass on either side. Figure 1121a. Like their lipid neighbors, these transmembrane proteins have both hydrophobic and hydrophilic regions. Their hydrophobic regions lie in the interior of the bilayer, nestled against the hydrophobic tails of the lipid molecules. Their hydrophilic regions are exposed to the aqueous environment on either side of the membrane. 2. Other membrane proteins are located entirely in the cytosol, associated with the inner leaflet of the lipid bilayer by an amphipathic alpha helix exposed on the surface of the protein. Figure 1121b. 3. Some proteins lie entirely outside the bilayer, on one side or the other, attached to the membrane only by one or more covalently attached lipid groups. Figure 1121c. 4. Yet other proteins are bound indirectly to one or the other face of the membrane, held in place only by their interactions with other membrane proteins. Figure 1121d. Proteins that are directly attached to a lipid bilayer, whether they are transmembrane, monolayer associated, or lipid linked, can be removed only by disrupting the bilayer with detergents, as discussed shortly. Such proteins are known as integral membrane proteins. The remaining membrane proteins are known as peripheral membrane proteins. They can be released from the membrane by more gentle extraction procedures that interfere with protein-protein interactions but leave the lipid bilayer intact. A polypeptide chain usually crosses the bilayer as an alpha helix. All membrane proteins have a unique orientation in the lipid bilayer, which is essential for their function. For a transmembrane receptor protein, for example, the part of the protein that receives a signal from the environment must be on the outside of the cell, and the part that passes along the signal must be in the cytosol. See Figure 1120. This orientation is a consequence of the way in which membrane proteins are synthesized, as discussed in Chapter 15. The portions of a transmembrane protein located on either side of the lipid bilayer are connected by specialized membrane-spanning segments of the polypeptide chain. See Figure 1121a. These segments, which run through the hydrophobic environment of the interior of the lipid bilayer, are composed largely of amino acids with hydrophobic side chains. Because these side chains cannot form favorable interactions with water molecules, they prefer the lipid environment, where no water is present. In contrast to the hydrophobic side chains, however, the peptide bonds that join the successive amino acids in a protein are normally polar, making the polypeptide backbone hydrophilic. Figure 1122. Because water is absent from the bilayer, atoms forming the backbone are driven to form hydrogen bonds with one another. Hydrogen bonding is maximized if the polypeptide chain forms a regular alpha helix, and so the great majority of the membrane-spanning segments of polypeptide chains traverse the bilayer as alpha helices. See Figure 410. In these membrane-spanning alpha helices, the hydrophobic side chains are exposed on the outside of the helix, where they contact the hydrophobic lipid tails, while atoms in the polypeptide backbone form hydrogen bonds with one another on the inside of the helix. Figure 1123. In many transmembrane proteins, the polypeptide chain crosses the membrane only once. See Figure 1121a. Many of these proteins are receptors for extracellular signals. Other transmembrane proteins form aqueous pores that allow water-soluble molecules to cross the membrane. Such pores cannot be formed by proteins with a single uniformly hydrophobic transmembrane alpha helix. The proteins that form pores are more complicated, usually possessing a series of alpha helices that cross the bilayer a number of times. See Figure 1121a. In many of these proteins, one or more of the transmembrane regions are formed from alpha helices that contain both hydrophobic and hydrophilic amino acid side chains. These amino acids tend to be arranged so that the hydrophobic side chains fall on one side of the helix, while the hydrophilic side chains are concentrated on the other side. In the hydrophobic environment of the lipid bilayer, alpha helices of this sort pack side by side in a ring, with the hydrophobic side chains exposed to the lipids of the membrane and the hydrophilic side chains forming the lining of a hydrophilic pore through the bilayer. Figure 1124. How such pores function in the selective transport of small water-soluble molecules across membranes is discussed in Chapter 12. Although the alpha helix is by far the most common form in which a polypeptide chain crosses a lipid bilayer, the polypeptide chain of some transmembrane proteins crosses the lipid bilayer as a beta sheet that is curved into a cylinder, forming an open-ended, keg-like structure called a beta barrel. As expected, the amino acid side chains that face the inside of the barrel and therefore line the aqueous channel are mostly hydrophilic, while those on the outside of the barrel, which contact the hydrophobic core of the lipid bilayer, are exclusively hydrophobic. The most striking example of a beta barrel structure is found in the porin proteins, which form large, water-filled pores in mitochondrial and bacterial membranes. Figure 1125. Mitochondria and some bacteria are surrounded by a double membrane, and porins allow the passage of nutrients and small ions across their outer membranes while preventing the entry of larger molecules such as antibiotics and toxins. Unlike alpha helices, beta barrels can form only wide channels, because there is a limit to how tightly the beta sheets can be curved to form the barrel. In this respect, a beta barrel is less versatile than a collection of alpha helices. Membrane proteins can be solubilized in detergents and purified. To understand a protein fully, one needs to know its structure in detail, and for membrane proteins, this presents special problems. Most biochemical procedures are designed for studying molecules dissolved in water or a simple solvent. Membrane proteins, however, are built to operate in an environment that is partly aqueous and partly fatty, and taking them out of this environment and purifying them while preserving their essential structure is no easy task. Before an individual protein can be studied in detail, it must be separated from all the other cell proteins. For most membrane proteins, the first step in this separation process involves solubilizing the membrane with agents that destroy the lipid bilayer by disrupting hydrophobic associations. The most widely used disruptive agents are detergents. Movie 11.3. These are small, amphipathic, lipid-like molecules that have both a hydrophilic and a hydrophobic region. Figure 1126. Detergents differ from membrane phospholipids in that they have only a single hydrophobic tail and consequently behave in a significantly different way. Because they have one tail, detergent molecules are shaped like cones. In water, they tend to aggregate into small clusters called micelles, rather than forming a bilayer as do the phospholipids, which are more cylindrical in shape. When mixed in great excess with membranes, the hydrophobic ends of detergent molecules bind to the membrane-spanning hydrophobic region of the transmembrane proteins as well as to the hydrophobic tails of the phospholipid molecules, thereby disrupting the lipid bilayer and separating the proteins from most of the phospholipids. Because the other end of the detergent molecule is hydrophilic, the association brings the membrane proteins into solution as protein detergent complexes. Figure 1127. At the same time, the detergent solubilizes the phospholipids. The protein detergent complexes can then be separated from one another and from the lipid detergent complexes by a technique such as polyacrylamide gel electrophoresis, discussed in Chapter 4. The complete structure is known for relatively few membrane proteins. For many years, much of what we knew about the structure of membrane proteins was learned by indirect means. The standard method for determining protein structure directly is X-ray crystallography. See Figure 448. but this requires ordered crystalline arrays of the molecule, and membrane proteins proved harder to crystallize than the soluble proteins that inhabit the cell cytosol or extracellular fluids. Nevertheless, with recent advances in crystallography, the X-ray structures of many membrane proteins have now been determined to high resolution, including bacteriodopsin and photosynthetic reaction center, membrane proteins that have important roles in the capture and use of energy from sunlight, an ability we review in Chapter 14. The structure of bacteriodopsin first revealed exactly how alpha helices cross the lipid bilayer, and the structure of the photosynthetic reaction center showed in detail how a set of different protein molecules can assemble to form a functional complex in a membrane. Bacteriodopsin is a small protein, about 250 amino acids, found in large amounts in the plasma membrane of an archaean called Halobacterium halobium that lives in salt marshes. Bacteriodopsin acts as a membrane transport protein that pumps H+ protons out of the bacterium. Pumping requires energy, and bacteriodopsin gets its energy directly from sunlight. Each bacteriodopsin molecule contains a single light-absorbing non-protein molecule called retinal that gives the protein and the bacterium a deep purple color. This small hydrophobic molecule is covalently attached to one of bacteriodopsin's 7 transmembrane alpha helices and lies in the plane of the lipid bilayer, entirely surrounded by the 7 alpha helices. Figure 1128. When retinal absorbs a photon of light, it changes its shape, and in doing so, it causes the protein embedded in the lipid bilayer to undergo a series of small conformational changes. These changes result in the transfer of one H+ from the retinal to the outside of the bacterium. The H+ moves across the bilayer along a pathway of strategically placed polar amino acid side chains. See Figure 1128. The retinal is then regenerated by taking up a H+ from the cytosol, returning the protein to its original conformation so that it can repeat the cycle. The overall outcome is the movement of one H+ from inside to outside the cell. In the presence of sunlight, thousands of bacteriodopsin molecules pump H+ out of the cell, generating a concentration gradient of H+ across the plasma membrane. The cell uses this proton gradient to store energy and then convert it into ATP, as we discuss in detail in Chapter 14. Bacteriodopsin is a type of transporter protein, a class of transmembrane proteins that move molecules and ions into and out of cells. See Figure 1120. We will meet other transporters in Chapter 12. The structure of a bacterial photosynthetic reaction center is shown in Figure 1129. It is a large complex composed of 4 protein subunits. 3 are transmembrane proteins. 2 of these, M and L, have multiple alpha helices passing through the lipid bilayer, while the other H has only 1. The 4th protein, cytochrome, is associated with the outer surface of the membrane, bound to the transmembrane protein. The entire protein complex serves as a molecular machine, taking in light energy absorbed by chlorophyll and producing high-energy electrons required for photosynthetic reactions, discussed in Chapter 14. Many membrane proteins are arranged in large complexes, and the structure of the photosynthetic reaction center reveals many design principles that apply to thousands of other membrane proteins whose structures are not yet known. The plasma membrane is reinforced by the cell cortex. A cell membrane by itself is extremely thin and fragile. It could require nearly 10,000 cell membranes laid on top of one another to achieve the thickness of this paper. Most cell membranes are therefore strengthened and supported by a framework of proteins attached to the membrane via transmembrane proteins. In particular, the shape of a cell and the mechanical properties of its plasma membrane are determined by a meshwork of fibrous proteins, called the cell cortex, that is attached to the cytosolic surface of the membrane. The cortex of human red blood cells is a relatively simple and regular structure, and is by far the best understood cell cortex. Red blood cells are small and have a distinctive flattened shape, figure 1130. The main component of their cortex is the protein spectrin, a long, thin, flexible rod about 100 nanometers in length. It forms a meshwork that provides support for the plasma membrane and maintains the cell's shape. The spectrin meshwork is connected to the membrane through intracellular attachment proteins that link the spectrin to specific transmembrane proteins. Figure 1131. The importance of this meshwork is seen in mice and humans that have genetic abnormalities in spectrin structure. These individuals are anemic. They have fewer red blood cells than normal, and the red cells they do have are spherical instead of flattened and are abnormally fragile. Proteins similar to spectrin and to its associated attachment proteins are present in the cortex of most animal cells, but the cortex in these cells is much more complex than that of red blood cells. While red blood cells need their cortex mainly to provide mechanical strength as they are pumped through blood vessels, other cells also need their cortex to allow them to change their shape actively and to move, as we discuss in Chapter 17. In addition, many cells use their cortical network to restrain the diffusion of proteins within the membrane, as we see next. Cells can restrict the movement of membrane proteins. Because a membrane is a two-dimensional fluid, many of its proteins, like its lipids, can move freely within the plane of the lipid bilayer. This diffusion can be neatly demonstrated by fusing a mouse cell with a human cell to form a double-sized hybrid cell and then monitoring the distribution of mouse and human plasma membrane proteins. At first, the mouse and human proteins are confined to their own halves of the newly formed hybrid cell, but within half an hour or so, the two sets of proteins become evenly mixed over the entire cell surface. Figure 1132. The picture of a membrane as a sea of lipid in which all proteins float freely is too simple, however. Cells have ways of confining particular plasma membrane proteins to localized areas within the bilayer, thereby creating functionally specialized regions, or membrane domains, on the cell or organelle surface. We describe some techniques for studying the movement of membrane proteins in How We Know, pages 382 to 383. As illustrated in Figure 1133, plasma membrane proteins can be linked to fixed structures outside the cell, for example, to molecules in the extracellular matrix, discussed in Chapter 20, or to relatively immobile structures inside the cell, especially to the cell cortex. See Figure 1131. Finally, cells can create barriers that restrict particular membrane components to one membrane domain. In epithelial cells that line the gut, for example, it is important that transport proteins involved in the uptake of nutrients from the gut be confined to the apical surface of the cell, the surface that faces the gut contents, and that other proteins involved in the transport of solutes out of the epithelial cell into the tissues and bloodstream can be confined to the basal and lateral surfaces. Figure 1134. This asymmetric distribution of membrane proteins is maintained by a barrier formed along the line where the cell is sealed to adjacent epithelial cells by a so-called tight junction. At this site, specialized junctional proteins form a continuous belt around the cell where it contacts its neighbors, creating a seal between adjacent cell membranes. See Figure 2023. Membrane proteins cannot diffuse past the junction. The cell surface is coated with carbohydrate. We saw earlier that many of the lipids in the outer layer of the plasma membrane have sugars covalently attached to them. The same is true for most of the proteins in the plasma membrane. The great majority of these proteins have short chains of sugars called oligosaccharides linked to them. They are called glycoproteins. Other membrane proteins have one or more long polysaccharide chains attached to them. They are called proteoglycans. All of the carbohydrates on the glycoproteins, proteoglycans, and glycolipids is located on one side of the membrane, the non-cytosolic side, where it forms a sugar coating called the carbohydrate layer. Figure 1135. By forming a layer of material covering the lipid bilayer, the carbohydrate layer helps to protect the cell surface from mechanical and chemical damage. As the oligosaccharides and polysaccharides in the carbohydrate layer absorb water, they give the cell a slimy surface. This coating helps motile cells, such as white blood cells, to squeeze through narrow spaces, and it prevents blood cells from sticking to one another or to the walls of blood vessels. Cell surface carbohydrates do more than just protect and lubricate the cell, however. They have an important role in cell-cell recognition and adhesion. Just as many proteins will recognize and bind to a particular site on another protein, some proteins called lectins are specialized to recognize particular oligosaccharide side chains and bind to them. The oligosaccharide side chains of glycoproteins and glycolipids, although short, typically fewer than 15 sugar units, are enormously diverse. Unlike polypeptide protein chains, in which the amino acids are all joined together linearly by identical peptide bonds. See Figure 1122. Sugars can be joined together in many different arrangements, often forming branched chains. See Panel 2.3, pages 68 to 69. Even 3 sugar groups can be put together in enough different combinations of covalent linkages that they can form hundreds of different trisaccharides. In a multicellular organism, the carbohydrate layer thus serves as a kind of distinctive clothing, like a police officer's uniform, that is characteristic of cells specialized for a particular function, and that is recognized by other cells with which each must interact. Specific oligosaccharides in the carbohydrate layer are involved, for example, in the recognition of an egg by a sperm, discussed in Chapter 19. They are also involved in our responses to infection. In the early stages of a bacterial infection, for instance, the carbohydrate on the surface of white blood cells called neutrophils is recognized by a lectin on the cells lining the blood vessels at the site of infection. This recognition causes the neutrophils to adhere to the blood vessels and then migrate from the bloodstream into the infected tissue, where they help remove the invading bacteria. Figure 1139. Glycoproteins are important members in the family of membrane proteins. In the next chapter, we further examine the sophisticated functions of transmembrane proteins that transport molecules into and out of the cell. Essential concepts. Cell membranes enable a cell to create barriers that confine particular molecules to specific compartments. Cell membranes consist of a continuous double layer, a bilayer of lipid molecules in which proteins are embedded. The lipid bilayer provides the basic structure and barrier function of all cell membranes. Membrane lipid molecules have both hydrophobic and hydrophilic regions. They assemble spontaneously into bilayers when placed in water, forming closed compartments that reseal if torn. There are three major classes of membrane lipid molecules: phospholipids, sterols, and glycolipids. The lipid bilayer is fluid, and individual lipid molecules are able to diffuse within their own monolayer. They do not, however, spontaneously flip from one monolayer to the other. The two layers of the plasma membrane have different lipid compositions, reflecting the different functions of the two faces of a cell membrane. Some cells adjust their membrane fluidity by modifying the lipid composition of their membranes. Membrane proteins are responsible for most of the functions of a membrane, such as the transport of small water-soluble molecules across the lipid bilayer. Transmembrane proteins extend across the lipid bilayer, usually as one or more alpha helices, but sometimes as a beta sheet curved into the form of a barrel. Other membrane proteins do not extend across the lipid bilayer, but are attached to one or the other side of the membrane, either by non-covalent association with other membrane proteins or by covalent attachment to lipids. Most cell membranes are supported by an attached framework of proteins. An example is the meshwork of fibrous proteins forming the cell cortex underneath the plasma membrane. Although many membrane proteins can diffuse rapidly in the plane of the membrane, cells have ways of confining proteins to specific membrane domains and of immobilizing particular proteins by attaching them to intracellular or extracellular macromolecules. Many of the proteins and some of the lipids exposed on the surface of cells have attached sugars, which help protect and lubricate the cell surface and are involved in cell-cell recognition.